scattered mixed clusters. And within clusters, what we see are two different gene structures. A standard gene structure, and where the promoter is in the middle, because this is a whole free gene. And then we have a gene that has an intron. So again, that's the standard structure for an intron in small genes. Remember, going back to that handout that you have on mRNA gene structure and organization, we have two different sets of symbols to show how we display introns and exons. So each one of these is transcribed. This one just needs to be trimmed because we have these transcribed spaces, and then folded. But this one we have a problem. We still have our transcribed spacers, and so we're still going to cut those. But now, when this one folds up, because of that intron, we have that intron. It's almost, not quite, but almost dead center in the middle of the gene, which puts it dead center in the middle of the anticodon loop. Remember, we did that on Monday. And so we can't just cut, because if we just cut away that intron, we're going to leave that anticodon loop open. So what we have to do is splice. So splice is a new term. Splice means that we have to cut and ligate. Actually, we're going to need two cuts. We have to cut there and there, and then we have to ligate those together. So we remove the intron, and we're left with something that looks like that. But it's a much more complex system. About a third to a half of your tRNA genes are split genes. So we talk about genes that have introns as split genes. The exons are split apart with an intron in between. And so we're going to start using that terminology now. And so this is a split gene, this is not. They're both processed by end trimming, and then we have to splice out the intron. Any questions on this? So the third of the three major RNAs in a cell, and again, there are probably 50 to 100 different minor RNAs, but we're only looking at the major RNAs. The third one is mRNA. This is the most complex. All ribosomal RNAs are identical. We only have about 40 different tRNAs. But every mRNA gene, virtually every mRNA gene, makes a different kind of a molecular protein. So we can't go through and give a general story for what mRNA genes look like. But you have that handout. You should bring that handout up to this chapter and continue to use that handout. That's literally mRNA gene structure and organization. But the way we're going to do this here is we're going to look at mRNA in bacteria, just general features of mRNA and how they compare between bacteria and eukaryotes. And then we're going to just pick out a couple of examples, because we're also doing regulation. We're going to pick out a couple of examples to go through the details of what a few specific mRNAs look like in bacteria and in eukaryotes. So this section is going to be very different from the section on ribosomal RNA and the section on tRNA. But I still advise you to make that spreadsheet and use that spreadsheet for the examples of mRNA that we're going to go through and compare that to ribosomal and tRNA. Is there any, do you have a question? Yeah.
Yes, did you go over gene regulation
Oh.
for tRNA?
Thank you. Very, very simple and straightforward. Exactly like ribosomal. And the reason for that is that the tRNAs, like ribosomal RNAs, are stable, and so you do not need to make new tRNAs unless a cell is divided. And so it's the exact same growth pathway, the signal transduction pathway that we've already looked at, that tells a cell it's going to duplicate everything. Anybody else? Yeah.
What was the gene regulation for bacteria?
The gene regulation for bacteria is also growth, but there it's ATP concentration. Very, very different. They don't have a signal transduction pathway like we do.
So they just make like one round of mRNA? Like a cell will only make like one round, and then when it divides?
No, absolutely not. So you'll make RNA as long as you need it. We're going to talk about how mRNA is regulated. And so it's very, very different, because every mRNA is regulated differently. And in fact, most mRNAs are regulated differently in different cells. So think about all the different cell types you have in your body. You're going to turn on muscle genes in muscle cells, but turn off muscle genes in nerve cells. That's the kind of thing we'll be looking at. Anybody else, before we look at some general features of mRNA in bacteria and in eukaryotes? So first of all, in bacteria, mRNA has coupled transcription, translation. And what that means is that as we are transcribing RNA polymerase, so we're making RNA, and while the bacterial genes are being transcribed into mRNA, ribosomes jump on and start translating and making protein. This is called coupled transcription translation. You do not do this because you have transcription in the nucleus and translation in the cytoplasm. So because you are doing these two functions in two separate locations in the cell, you can't couple them together. And so this allows bacteria to do some kinds of regulation that you can't do. It gives bacteria an advantage in some some ways because it can regulate some things that you can't do because you have to wait for a message to go out into the cytoplasm. Yes?
Alright. In the case of either the European says or as transcribing RNA poly occurs,
Just what you see. As transcription occurs, translation is happening also.
the ribosome is also translating.
The ribosome is also translating during transcription. Yes.
What is the lettering above the DNA there? Is that RNA?
RNA polymerase, RNP. Any questions on what is coupled transcription translation? You don't do it. Bacteria is polycystic. You've already seen that on the handout that you already have. That handout that I gave you weeks ago shows polycystic message is the norm, but not exclusively in bacteria, and is extremely rare in eukaryotes. For all practical purposes, you can say eukaryotes do not have polycystic messenger RNA. It's that rare. Any questions on what I mean by that? So this is 1, 2, 3 is the half-life. So the symbol for half-life is T1/2. Same definition if you're talking about the half-life of a radioactive element or the half-life of a messenger RNA in the cell. A half-life means how long does it take for 50% of the material to disappear. That's what the half-life of radioactivity is. How long does it take for half of the atoms to disintegrate? Here we're talking about how long does it take for 50% of the messenger RNA to go away. In bacteria, half-life is measured in minutes. You measure half-life typically in hours. You do have some messenger RNAs with short half-life, but they're unusual. The reality is that half-life, that messenger RNA decays so fast that the messenger RNA starts to decay while it's being made, while it's being translated. So what that means, the impact of that is that if a bacteria changes its mind and wants to stop making the enzyme that this gene codes for, all it has to do is stop transcribing the gene. So that puts the decision of which proteins to make almost exclusively at the level of transcription. Because you make messenger RNAs that last for hours and sometimes days, that means that if you want to stop having that enzyme around, you've got to do special stuff in the cytoplasm to get rid of proteins. And so in terms of the big picture of gene expression, there are pluses and minuses to having these two systems. Bacteria doesn't have to worry about messenger RNA outliving its usefulness. You have to have some mechanism to consider messenger RNA that may no longer be needed. And so that changes the dynamics of gene expression. Any questions on these three things? Yes?
What is polycystic defined as again? So you can see right there.
Means multiple cistrons in a transcription unit. Polycystron. multiple cistrons and a transcription unit. Anybody else? So number 4 is you modify your message. Bacteria do not. Yeah, you can see that. Any questions? Anything on this? I want to erase this. So bacteria use the message exactly as you see it. No modification at all. Right off the template, ribosomes jump on. You do modify your message, and so we're going to talk about 4 different kinds of modification to your message. Basically, but not entirely, these modifications have to do with the fact that your message, in most cases, has to last hours in the cell. So most, but not all of these modifications have to do with protecting the message against decay. So we have a general pattern, a general picture of what a eukaryote message looks like. It starts at the 5', ends at the 3'. The reality is, because your message has to last hours in the cytoplasm, we're going to protect it with proteins. We've done this over and over again. I think the first time you saw that was in replication. We had single-stranded DNA binding proteins, and so now we have single-stranded RNA. So we have to protect the mRNA with proteins. This is not like replication, however. It's not a single protein, but rather there are multiple families of proteins. So we'll just say proteins in general. And what that does now is we have this complex of RNA and protein. This is now called an mRNP. P now stands for protein, a messenger ribonucleoprotein complex. And so all mRNA is complex with proteins in the nucleus, on the way out to the nuclear pores, in the cytoplasm, and even while being translated by ribosomes. Remember, bacteria is not going to do any of this stuff that we're talking about. Any questions on what I mean by mRNP? Next thing is, at the 5' end, we're going to add a cap. So again, for the same reason, we have to protect the message so that nucleases can't get in and destroy it. So a cap just caps the end of the message and prevents enzymes from getting into the message. So a normal messenger RNA messenger RNA almost always starts with A. And ATP is the first nucleotide that the RNA polymerase puts in. And so a messenger RNA, or actually any RNA, tRNA, ribosomal RNA is the same, any RNA still that's what ATP is. So we're going to cap this. And so the cap is a backwards guanosine nucleotide. That's the cap structure. Notice how bizarre this is. We are talking about a 5' triphosphate linkage. So when I just write cap here on the 5' end, this is what cap is. There's a capping enzyme that puts on this backwards guanosine nucleotide in a triphosphate linkage. So typically, we have a phosphodiester bond. Now we have a triphosphate bond linking the cap to the body of the message. Everybody see what that is? Any questions on what cap is? Now we come to introns. The vast majority, not all of your messages, have introns. You all know that. You had a homework problem. You saw some real genes. You have that handout. It has your average gene with 4 or 5 introns, little, tiny exons, big, giant introns is your pattern for how your genes look. But whether it's your genes or Drosophila with a moderate amount or yeast with very few, splicing nuclear message is identical from yeast all the way up to deviants, including plants. And so what we have this symbol that we've been using for splicing is all about making cuts. So we cut, and then we ligate this end to that end. That's what that V designates. That's the standard system for splicing nuclear messenger RNA. Now, the splicing of nuclear messenger RNA is autocatalytic. Autocatalytic means that the RNA splices itself. We are not talking about an enzyme, a protein enzyme that comes in and splices. We did have that, and it's gone. We did have that for the tRNA. Where a protein enzyme makes the cuts and another enzyme ligates it. This is completely different, totally 100% different than that picture I had on the board for tRNA. 100% different. The intron itself splices itself out. That means that this intron is called a ribozyme. So a ribozyme is an RNA enzyme. That's what this is short for. which makes the two cuts and stitches together the two ends of the exon. So this is a brand new word. You're used to having proteins with enzymatic function, and now I'm telling you that RNAs, in lots of cases, have RNA function. We're going to see this again. RNAs, in some cases, have the ability to act as an enzyme. Now, for this RNA splicing, proteins are involved, but it's the same conundrum that people face with the chromosome. Chromosome was DNA and protein. They thought the protein was the gene. Well, the splicing factors are proteins, and people assume that they were doing the splicing. All the proteins do is fold up the intron. So if you're into this system, the proteins are playing no active role other than folding. You had a question?
Yeah, the ribozyme, is that what's actually doing the splicing?
Yes. So the term ribozyme just tells you that the intron is acting as an enzyme splicing itself.
Okay.
So anytime you see the word ribozyme, that means that the function is carried out by an RNA acting as an enzyme. That's what that means.
Where then does a splicezyme work?
Splicezyme are a set of proteins and RNAs that are involved in folding this up. And because every intron has a different sequence, there are a few general sequences. There's more details in your textbook that I don't want you to learn from my exam. And so that will show you how the intron is folded up by a combination of RNAs and proteins which have absolutely no catalytic function. The spliceosome is only there for folding. It is the intron RNA itself which cuts itself and ligates the two exons back itself. So the details are in your book. You do not need them for the coming exam. Anybody else? Yeah.
I'm sorry, so is it on the cap structure? What does the G stand for?
Guanine. It starts off as a GTP. So the capping enzyme takes a GTP and puts it on backwards.
Okay, and what's the
Oh, I'm sorry, that's methyl. So the guanine is methylated. I forgot to say that. I'm sorry. Yes. Why? Just because? It just is? I have a really hard time with why questions. Yes?
What's to stop an enzyme from going in the 3' and destroying the sequence? But it just has to be, the enzyme also has to be active.
I said there are four things. And so we're going to cover that. We're going to cover the front, the inside, and the back end. Yep. Good point. Yes?
Where does the ribozyme splice itself out?
So this is exactly where I'm pointing.
Oh no, like in the nucleus and the cytoplasm or
This is
wherever.
all nuclear.
Okay.
Yeah, so all of this is nuclear. It all happens, actually it happens during transcription. So the bacteria is transcription translation. The eukaryote stuff is transcription processing happening at the same time. So as the RNA is coming out, as the polymerase is going down the gene, it gets capped first, protected, spliced, 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 and then we're going to protect the back end. Anybody else? Any questions on this so far? Okay, so at the back end, we're going to put what's called a poly A tail is released from the DNA, an enzyme complex comes along and adds a tail and extends it out to about 200 nucleotides. So that means that if an enzyme happens to grab onto the end of the message, it's not grabbing on to the transcribed part, it's grabbing on there, and in the cytoplasm, there's repair enzymes, and so in the cytoplasm, the tail is lengthened, shortened, lengthened, shortened, lengthened, shortened, until it gets a signal we don't need it anymore, and then it just gets destroyed. And so, from one end to the other, your messages, with one exception, have these 4 protections to prevent degradation. The only exception are the histone messages. So remember the histones that code for the nucleosome. Those messenger RNAs do not have a poly-A tail. They are capped, they actually aren't spliced either. They're capped and protected with proteins. But they're very, very special. All your other messages have all 4 of these. Any questions on that? Yes?
So, the poly-A tail, what exactly does it do?
It protects the end, the 3' end of the message, and as we'll talk about very briefly from the handout, it also aids in translation. Oddly enough, even though it's at the end. Anybody else? Any last questions before I swap out the boards? Okay. So, we now have all of the components necessary for translation. Translation means protein synthesis. We know the ribosome is the factory. We know tRNAs decode. We know mRNA is the blueprint to make the protein. This all happens on the ribosome. The protein synthesis happens on the ribosome. We have our messenger RNA, and I'm going to do this generically because at this point it doesn't matter whether we're talking about bacteria or whether we're talking about humans. We have a messenger RNA, 5' to 3', just like I have up there. I'm just not putting the cap on or the tail on. Messenger RNA is read 5' to 3'. So translation is 5' to 3'. So pretty much almost everything that happens molecularly happens 5' to 3'. All of the key processes, replication is 5' to 3'. Transcription is 5' to 3'. And now you're seeing that translation is 5' to 3' also. So you know what that means. So the building blocks for translation are amino acids. And I'm going to continue to abbreviate it AA because I'm lazy. So the amino acids are the building block for translation. That's an outline of a generic amino acid. So a generic amino acid amino NH2 group, a central carbon, and a carboxy terminus. So just like a nucleotide, it has two different ends. This is a three-part molecule. From the central part, the carbon, there are 20 different R groups. Each R group is specific for a different amino acid. It can be as small as a hydrogen or as large as a ring compound. So this is the equivalent, this is the building block equivalent to a nucleotide. A nucleotide builds a chain of RNA or DNA. An amino acid is the building block that builds protein.
So the R on the bottom, is that going to be like lysine, methionine?
Yeah, right. So all your 20 amino acids, the 20 different amino acids have 20 different R's. And I'm not going to get into any of that. You can hold that off for biochemistry. We're not going to play with that at all. Just R is fine. Anybody else?
At the beginning of this, is that supposed to be,
NH2. NH2. So I just wrote it out. You'll see why in a second. Anybody else? All right. So just as with nucleotides, nucleotides are put together by a polymerase, either a DNA polymerase or an RNA polymerase. I changed the back end of that amino acid. It means exactly the same thing. I have to write out the middle one for a reason. And so what the ribosome does is it takes OH from the first amino acid, the H from the second, and a water elimination H2O, and we end up with a peptide bond. So a peptide bond is the equivalent of the phosphodiester bond in nucleic acids. The peptide bond links adjacent amino acids in your protein. It is the functional equivalent, but a much simpler, it's not a di bond, like a diester bond in nucleic acids, but it is the link that allows the ribosome to polymerize amino acids into a polymeric protein. Any questions so far? Polymeric protein. Protein is a polymer of amino acids. DNA is a polymer of nucleotides. RNA is a polymer of nucleotides. Any questions on what is, so the peptide bond that is the peptide bond. Any questions so far? Biochemist can kick back and switch. Okay, now, just as with a nucleotide chain that has a distinct and different 5' end and a different 3' end, a polypeptide or protein has two different ends. So this is an amino group. This is called the amino end, or typically just N. Means the exact same thing, totally identical. At the back end, we have the carboxy end, or the C end. Simple, straightforward. So translation occurs, so we have here 5' and 3', and at the same time, N to C. So 5' to 3', transcription is, the message is then followed by amino to carboxy translation. So that is functionally the same. So we have a gene with an open reading frame. AUG to stop is transcribed 5' to 3', giving a message which is translated N to C. The relationship between transcription and translation. Any questions on this? Everybody see what's happening? I did not say that. I will not tell you yet. It's a surprise, actually. Any questions not on the details of how protein synthesis, that's in the handout, we're going to get to it. Maybe not today, we'll get to it. But on the connection between transcription and translation. In it's already gone away. In bacteria, they're coupled. You do transcription in the nucleus, translation in the cytoplasm. Any last questions on this part? Okay, let's do an in-class cooperative learning exercise. This is a real good one. When you see it, you may recognize it as one of the problems on the homework that's due on Friday.
Sorry.
That's on Friday. Where it's 12:50. Or is your phone based
It was 150 where
Well, daylight saving time just happened.
Yeah, but then I corrected it already. And
in Arizona.
Arizona doesn't change.
I know. Well, the iPhone doesn't know if it didn't change sometimes. That line.
Okay,
Mine
hold
said
on
the general
for a
melting
few
point.
minutes. I have to give you instructions for this.
I mean, it was working like literally before class. It's really weird.
What you're being asked to do here is to translate a segment of DNA into protein. So that means that you've got double-stranded DNA you're going to translate a segment of DNA, you have to go through the process of transcription and then translation. So 5 prime, 3 prime, 3 prime, 5 prime, anti-parallel means that the transcript, so that's the RNA, 5 prime, 3 prime, 5 prime, 3 prime. And since you're being asked to find an open reading frame, that means that one of these molecules has to start with AUG and end with one of the stop codons. I put that up backwards. I don't like to write backwards. So it's got to be AUG or AUG UAA. So that's your task for this, is to take that double-stranded sequence, transcribe it into an RNA, just conceptually, don't write it all out until you figure out which one. Now it says, what does the open reading frame code for? Well, the open reading frame has to start with AUG. The easy way to find the start of translation is to look for the cat. You hear that? Why cat? Why are you going to look for a cat?
Because it's the GUA and then you read backwards is the AUG.
It's the easiest thing in the world. As you skim sequence, DNA sequence, not RNA because there's no T, as you skim DNA sequence, your eye just automatically, once you have the word cat in your head, your eye just locks on, doesn't matter whether it's forwards or backwards. So what you want to do is look for the cat, make sure it's in the right direction, and you've got the code dictionary. Again, I'm not going to make you memorize it. You've got the code dictionary in front of you. Just translate it. This turns out to be a very, very simple project. But it's setting you up so you can now do the last homework problem that's due on Friday. So now, using those directions, go ahead, find the open reading frame. What does it code for? Decode the open reading frame. Not a problem.
You have to read that one, don't you? Right here.
Yeah, no, okay, so,
No, that's true. It's not.
So then you would read it backwards, so you wouldn't start with med.
Every time you get like a new step, you're going
Why?
Because when you see it, actually, see how it says cat when it's read backwards already? And it's going from the RNAs 5 that's on your RNA.
So you would have to read it, right?
So you read it 500. So
Yeah. Yeah, so AUG, and the next one is CD. No, it's CD. Because you have to keep going backwards. Yeah, so you probably put, yeah, met is always first. Okay. So it's read by three, not translated. No, it's C.
Don't ever say stop.
don't ever say stop. Don't ever stop.
Is it on?
Are they online?
It should be on mine. I'll check when I get back.
Yeah, I would have never done it.
Then he should have.
Okay, shut it. Shut it.
We're doing Jeopardy in one of our classes. First, 6, and 7, Suck it long, suck it hard.
I'll take the rapist.
It's like the rapist for 500. I'll take Japan relations. That's Japan, US relations, and never mind. We're just gonna take S words for 500. Number 7. This is a category where the answer to everything is 7. Ah. Do you get it? So yeah, there'd be in front of that A. I wouldn't do it on a test. I wouldn't chance anything. I would literally verbatim just write this. Oh, like when you write? No, unless, I mean, I guess this is easier if you were Japanese. What's up, yo? Oh.
I guess you're like, okay, good man. So,
The ceiling? I haven't gone and looked yet. I'm afraid to, because everybody I know says that they've done really bad on the exam. I'm afraid
you
to.
might want to look. Maybe you got there. Maybe you did real bad.
I'm afraid to look.
Alright.
I don't even know where his office is. I haven't picked up any of my books.
It's really weirdly located. It's like underground in the basement of the supply chain. Yeah. Like before you get there, there's like doors that bring something to the floor on it. I think it's just a good bios.
I have to like have someone go with me just to show me where it is.
but that involves printing.
That's really an idea myself.
Print it out though.
Yeah, but then how am I going to take it with me? You're making this. I'm just going to say it's one button. If that doesn't like take up time. What's up? You got methionine, arginine, and phenolphthalein. I don't know what phe stands for.
Class be over now.
13 minutes. No, 18 minutes.
Yep. I was asking. No, that's not it. That's fine. Especially as it makes it easier for you to run.
Med arg phe.
Okay, that didn't take too long, you guys did a good job. Once you get the hang of it, this kind of problem becomes very simple. But there's some very important rules that you need to know to avoid losing points. So first of all, and most importantly, there is no amino acid called stop. So when you're asked what is it code for, so coding cropped up on the last exam, and a lot of people just wrote a nucleic acid sequence. Nucleic acid sequence isn't coding, it's what it is, but it codes for an amino acid. So what does it code for is amino acids. Now, when you do that, because this problem happens to be set up going from right to left, you can write the amino acids down, but if you don't turn it around, that's fine. Just put an N in front of the met. And that tells me that you know the methionine is the N-terminal or beginning of that polypeptide. So, number one, never write stop when you're asked for coding. Number two, if you write the amino acid polypeptide backwards, just put an N in there to tell us that you know it backwards, that you didn't have time or whatever to turn it around. Those are really important when you're decoding a section of DNA. So this was a really simple one just to get you started. When you look at the homework, there'll be a few cat boxes in there deliberately. And so what you have to do is evaluate each one. So the problem is worded the same. What does it code for? And one open reading frame. You know the definition of an open reading frame is a start codon to a stop codon. And so you always start with methionine, and you always end with whatever the codon is immediately before the stop codon. So these problems are really, really simple once you get the hang of it. Any last questions on how to do this in-class problem? Yes?
So if you write N at the start.
Well, only if you're writing it backwards. Because this one was set up backwards, if you write it backwards, you have to tell me that you know that the methionine is the start, and that just means putting an N, because
Why do we have to write N? Sorry.
because it's the amino terminus.
Okay.
It's the amino, so in the real proteins, just like you write 5' and 3', protein has an N and a CN. And so if you write it backwards, I have no way of knowing if you know it backwards unless you tell me what the amino N is.
Oh, N, not N. Okay.
N. Yes. Everybody clear on that? So half of you wrote it backwards, and that's okay. Just make sure you put an N, the letter N, in front of the methionine. That tells me you know that it's really reading backwards, and you didn't have time to change it. The homework, that shouldn't be a problem, you have lots of time. Yes?
Should we put the carboxyl end in there too, or no?
That's not necessary. Any last questions on this? Yes?
What about a C like for a
Not necessary. Just the N is the most important. because it's where you start that is most important. Anybody else? Okay, so let's look at that handout. First of all, is the handout protein synthesis in a eukaryote or a prokaryote? How do you know? It's got a nucleus. What's the second way that you know this is in a eukaryote?
The number.
What number? 40S and 60S, the ribosomal subunits. Now, as you look at this, so we're going to do this in bacteria. The mechanism is virtually exactly the same. There are a couple of little interesting differences that we may or may not talk about, but the outline is exactly the same. Notice that we're starting off on the upper right-hand side. And so the first thing that we have to do to start translation, to start protein synthesis, is to put the amino acids on every tRNA. That term is acylate. And so we have this tRNA. This is a plain tRNA. If we acylate this, and we have covalently joined an amino acid to the tRNA. There are 20 standard amino acids in your dictionary. There are 20 different enzymes that acylate its family of tRNAs. Some of them are really simple. There's only one codon for methionine, but look at all the codons for leucine. And so a given enzyme called a synthetase can acylate multiple transfer RNAs. Now we have a standard set of vocabulary for talking about transfer RNAs and the symbols. So you know the symbol of tRNA, that's just any generic transfer RNA. You know that we start off translation with AUG, which codes for methionine. The way that I tell you that this is a tRNA for methionine is by a trailing superscript. So writing net at the end on top tells you that this transfer RNA, this one, now this one, not just amino acid, but specifically methionine. Most of the amino acids have multiple transfer RNAs because of all the codons. Even though methionine only has a single codon, there are two different tRNAs. When I say different tRNAs, because it's methionine and because the codons can only be AUG, that means that the body, the sequence of RNA in here is different. So in general, we specify different amino acids by a trailing subscript. So we have tRNA net F, and we have tRNA net M. Two different tRNAs, different sequence in the body, but the same decoding as AUG, CAU in the tRNA. They are exactly the same. The difference, the reason that we have net F and net M, this one is used to initiate only. That is the start AUG codon. This one is used in case there is an AUG internal. So methionine is a plain ordinary amino acid that just happens to be used to start. And so lots of proteins have methionine inside the protein. And we have two different transfer RNAs. You'll see the way that plays out on Friday when we actually start to go through the handout. So be sure to bring this handout back on Friday. So this is just this. Now, once we put an amino acid on methionine, so writing methionine in front of the tRNA now tells you that this transfer RNA is acylated. This transfer RNA now has an amino acid attached to it, and it's attached to the right, the correct transfer RNA. So the set of symbols that we use when we have an amino acid in front of tRNA, that tells you what amino acid has been covalently attached to the transfer RNA. When we're writing it as a superscript, it tells you what the anticodon is. And M and F distinguishes different forms for the same amino acid. But remember now, we have different codons for some amino acids, and so there's a difference there also. So that's the symbology that we are going to use. Any questions on that? Yes?
For the other amino acids, do they have an F subscript?
No, so most of the other amino acids are going to use 1, 2, 3, 4, 5, 6. So M and F is unique, only used for methionine. Anybody else? Any questions on the terminology here? So let's take a quick look and just start things off. First of all, what you see is that translation does not start with a ribosome. In fact, if you see a ribosome, so that's a ribosome. If you see that in a cell, it's just an inactive factory. All cells have a surplus of factories, just in case they have to ramp up translation. Translation only occurs on what is called a polysome. So polysome is the term that is an abbreviation for a polyribosome, a set or group of ribosomes translating a message. So 5' to 3' on the message. This one, we are moving in this direction. That means that number 1 is the one that entered the message most recently. Number 3 is a ribosome that's almost done translating the message. So it has a protein hanging down. Number 2 isn't as far along. It has a shorter protein. Number 1 just started. It has a very short protein. So each ribosome jumps on at the 5' end and then works its way. Translation, remember, is 5' to 3'. Works its way down until it hits the stop codon. And so each ribosome will transit the entire open reading frame. But this is polycistronic. There's a couple of different things here, but we're not going to worry about that. And so this will keep going until it hits a stop codon and then release the protein. That's the big picture for what we are going to do.
Each ribosome is coding for the same protein.
Yeah, so each ribosome here is making exactly the same protein. The bigger the message, the more ribosomes on the message. Some messages are very large, some are very small. Each one is making the same one. It's going to go down until it hits the stop codon, release its protein, and then look for another cap or start of translation.
So it'll not read the tail at all. It won't make that poly A tail at all.
Oh, no. So this is generic. So I don't have a cap or a polyetail. This is identical in bacteria. There was a hand up over there? Yes.
They say 1, 2, and 3.
Yes.
So why would we not put 1 on the first?
Yeah, you're right. Thank you. I just labeled them generic. Anybody else? Make sure you bring this handout back on Friday.